Hello everyone, this is Dr. Santosh. Welcome to Dr. Santosh Medhavi University. So today we'll be talking about a very important topic, VLDL, IDL and LDL metabolism. In this topic we'll be talking about very essential questions. So please stick with the video and don't forget to like the video, subscribe the channel and press the bell button so that you'll get notification. Let's start with the today's topic. So we'll today talk about VLDL, IDL and LDL metabolism. So VLDL actually converts into IDL and IDL converts into LDL. So actually these are the integral protein wise, these are VLDL, IDL and LDL both have same integral protein, that is B100, and that is why LDL will convert into IDL and IDL will be converted into LDL and LDL will be taken up by LDL and IDL both can be taken up by hepatocytes. Now let's this break this process of VLDL metabolism into 6 parts so that we'll understand this very easily. Now first step here is assembly step. So and second is secretion, third is maturation, fourth is distribution, fifth is apoprotein shedding, and sixth is uptake of lipoprotein. First step, if we can see in first step, what happens is the lipid in hepatocytes, lipid is being synthesized. Both fatty acid is synthesized and glycerol is synthesized. So fatty acid and glycerol react to form triglyceride, and that those triglyceride will be loaded to B100 protein, and this loading is done in microsome, that is endoplasmic reticulum, by the protein called microsomal triglyceride transfer protein, MTTP, microsomal triglyceride transfer protein. This is very important protein, and the same protein is involved in formation of chylomicron in enterocyte. This same protein is functioning as assemble. The same protein will load the triglyceride in hepatocyte also, the MTTP protein. So if there is deficiency or mutation in MTTP protein, this will lead to A beta lipoproteinemia. This is very important disorder in lipoprotein metabolism. What happens in this A beta lipoproteinemia is in blood there is no B100 or there is no B48. So there is no chylomicron VLDL, IDL and LDL in blood. So what will happen is, if there is no V48 chylomicron, so lipid will not be absorbed from the intestine and lipid will not be transported across the tissues. Because of that, there will be deficiency of fat soluble vitamins and essential lipids, and this will cause big problem. So this is very important, one of the very important disorder. Understanding of this single disorder will make you understand many things. What is the function of this essential fatty acid? You will understand if you study this other. What are the function of this fat soluble vitamins? You will understand how this fat soluble vitamin defect present in the patient. You will understand those all things. You actually understand the significance of fat in our body. Now let's come to the second step. So what will happen is, if second step, if there is in A beta lipoprotein, if there is no MTTP, so what will happen? There is no B48, there is no assembly step, there is no assembly of lipid into this B48 or B100, and if there is no assembly, there will be no secretion, and if there is no secretion, there will not be B48 B100 inside bloodstream. And this secretion after assembly occurs through exocytosis. Remember, secretion through after assembly occurs through exocytosis, and assembly occurs in endoplasmic reticulum, and secretion, after secretion, this VLDL is directly secreted into the bloodstream, while chylomicron, as we have talked before, it is secreted first in lymphatics. After lymphatics, it will reach into bloodstream, but VLDL from liver directly secreted into bloodstream. Now third step is maturation step. In maturation, this VLDL is nascent VLDL, secreted VLDL is nascent VLDL, but this nascent VLDL will get mature. What does maturation means? This VLDL will be capable of metabolism. its own metabolism. So this is called mature VLDL. So maturation takes place after receiving this apoC II and apoE protein from HDL, because HDL is the source of all peripheral protein. So this nascent VLDL will get apoE and apoC II and mature into mature VLDL. Now fourth step is distribution. After this VLDL, nascent VLDL convert into mature VLDL, there will be rapid distribution of lipid around the tissues, adipose tissue, muscle and cardiac tissues. So there will be rapidly distribution of this triglyceride and other lipids around the tissues. And after distribution, so this enzyme plays very important role, very important enzyme. The deficiency of this enzyme will result in type 1 chylomicronemia. So this is very important enzyme, lipoprotein lipase enzyme, and this lipoprotein lipase enzyme will help in distribution of lipids of VLDL. And lipoprotein lipase form one complex with C2. C2 is peripheral protein that is present on the VLDL, and that this C2 comes in VLDL from HDL. So lipoprotein is activated by C2 that is present in VLDL, and this form complex, lipoprotein lipase C2 complex. So lipoprotein lipase and C2 in combination, they will catalyze the VLDL. It converts the triglyceride inside the VLDL into glycerol and fatty acid. Glycerol and fatty acids are transported across the tissue, and tissue will, either adipose tissue will store those glycerol, fatty acid, and other tissue will metabolize. So this lipoprotein lipase disorder, or if there is disorder of this C2, it will cause type 1 hyperlipidemia. Type 1 hyperlipidemia is also called chylomicronemia. How do you remember? You can see this one, you can see L and L. So you can put this L, and there are also one type of L or one type of structure. So L type of straight line, vertical straight line repeats for type 1. So 1 is there. Now how do you memorize this type 3? Type 1 is there, then type 3 is because of apoE. Defective apoE. So defective apoE will cause hypertriglyceridemia. apoE means type 3. So you can see this E is very similar to 3. So apoE means type 3. So this is a funny correlation, but it will help you. Though this type of hyperlipidemia is written in Roman letter, I have written this 3 because so that you will correlate this E with 3. So apoE defect will cause type 3, and type 3 means tri. So apoE, type 3, and hypertriglyceridemia. So you can remember. Now type 1, chylomicronemia. Chylomicron is the first lipid, the largest lipid, and that is why chylomicronemia type 1. Now last step is uptake, and this is related to that. So first, going to last step, let's talk about this apoprotein shedding. So at this step, what will happen? I have not written any protein, because what will happen is VLDL will give peripheral protein back to HDL. So peripheral protein, especially this C2, will be given first, and then apoE1 will be given. If this VLDL gives C2 first, VLDL will convert into IDL. When IDL give apoE, then it converts into LDL. LDL only has B100 and lipid in it. Which lipid? Lipid cholesterol in it. Now this LDL will taken up by LDL receptor of hepatocytes. Generally 70% of LDL will be taken up by hepatocytes. So LDL receptor hepatocytes, and this LDL receptor will take up this LDL, and there is IDL will also be taken up by LDL receptor related protein, mainly LDL receptor related protein. And this, when there is defect in this LDL receptor, it will cause familial this is also called type 2 hyperlipidemia, is also called familial hypercholesterolemia. So this deficiency or defect in LDL receptor or low number of LDL receptor will cause type 2 hyperlipidemia, and that is called familial hypercholesterolemia. You can see this receptor is related to type 2. So you can write receptor like R. Remember, this is 1 and you can write it 2. So receptor is 2. So LDL receptor is related to type 2 hyperlipidemia, and type 2 hyperlipidemia is familial hypercholesterolemia. So LDL receptor is for type 2 hyperlipidemia, and type 2 hyperlipidemia means familial hypercholesterolemia. These are very, very important points that you have to remember for your exam, and for USMLE also. These are very, very important. Hyperlipoproteinemia, if you understand this, actually you will grab the concept, basics of this cause of this all disease. If you have the basic of cause, you'll understand whole thing. The cause is the root, so you'll understand whole manifestation of this disease easily. Now let's come to the other slide. We'll talk about how this VLDL gets metabolized, how this VLDL is assembled, how is this VLDL secreted, and then maturation happens, then distribution, then apoprotein shedding, then uptake. This all process we will understand through the picture. Let's say this is the picture showing VLDL metabolism. VLDL assembly, you can see here VLDL assembly, then second step is secretion from liver to bloodstream, then after that maturation. So this VLDL will accept apoprotein from HDL and it will convert into mature VLDL, then mature VLDL will convert into IDL, and then IDL will convert into LDL. Now let's see how this metabolism happens. So first, this assembly occurs. This assembly is done by MTTP protein. So MTTP protein is microsomal triglyceride transfer protein. This protein is present in endoplasmic reticulum of hepatocytes. This protein is also present in endoplasmic reticulum of enterocytes, and we are talking about VLDL. VLDL is secreted from hepatocytes. Now see what will happen is in hepatocytes, this lipids, hepatocytes, this all process is happening during fat condition. Remember this very important concept. This whole concept is predominantly happening during fat condition. In fat condition, this VLDL synthesis is boosted up. So what will happen during fat condition? After we eat food, so excess carbohydrate or lipid that are there, they will convert into fatty acids, and this fatty acids that is there, so fatty acids and triglyceride, they will assemble into triglyceride. So fatty acid synthesis will synthesize this fatty acid in liver, and glycerol synthesis will form glycerol 3-phosphate, and they will assemble, and after assembly they will form triglyceride, and this triglyceride and this endoplasmic reticulum of hepatocyte also produce B100 protein. This is B100 protein, though I have not written anywhere. This is B100 protein, which is integral protein of VLDL, IDL and LDL, and this B100 protein, on B100 protein, these triglycerides are loaded. Some cholesterol will also be there, but not that much cholesterol. Some cholesterol, other lipid, but predominantly triglyceride will be loaded here. After loading this triglyceride, this ApoB100 will transform into the particle called ApoB100. So many ApoB100 particle will assemble in vesicle, and that vesicle actually whole process is happening inside the endoplasmic reticulum. Now from endoplasmic reticulum what will happen? So ultimately vesicle formation happen. That vesicle will contain many VLDL, and those VLDL will be secreted through exocytosis. After secretion of this VLDL particle through exocytosis, this VLDL is not capable for metabolism. So what will happen? This nascent VLDL will mature. And how does it mature? It mature, second phase is maturation. It mature after receiving these two apoprotein C2 and apoprotein E. And after maturation, what will happen? This is capable of distribution of lipid. This is capable for metabolism. Now, so this C2, this is C2. This is C2, and this is E. So what happens is C2 will interact with this very important enzyme, and that is present on the endothelial surface of different tissues. So imagine this reaction is happening. If you see, if just imagine the context, what will happen is when heart pumps the blood, so blood through arteries, through arterioles, it will enter into the capillary. If it has to enter into tissue, so tissue will have various tiny capillaries. In those capillaries, this enzyme is present attached with the endothelium membrane of this capillary through this carbohydrate called heparan sulfate, and this very important enzyme is called lipoprotein lipase. And as I've told you, you must remember this whole process is happening during fed condition. In fed condition, what will happen is lipoprotein lipase concentration will increase because of insulin, and this is insulin's friend. This lipoprotein lipase is insulin's friend, especially in adipocytes. So insulin causes increased synthesis of this lipoprotein lipase, and increased lipoprotein lipase at adipocytes will cause this lysis of, actually breakdown of triglyceride into glycerol and fatty acid, and this glycerol and fatty acid is taken up into adipocytes. In adipocyte it will assemble and will be stored. But what about this cardiac muscles, other muscles and cardiac muscles, especially cardiac muscle? What will happen? This cardiac muscle will take this fatty acid and it will convert into energy. They does not store, they convert that fatty acid into energy, but in adipocyte they will be stored. Now after distribution, this VLDL, mature VLDL will be very rapidly distributed. Okay, this VLDL will be distributed rapidly, and then after distribution what will happen is this VLDL will give its C2 apoC2 protein back to shed its own C2 protein back to HDL. After shedding, this is fifth step, is shedding step. Fourth step, distribution. After distribution, shedding step. So after shedding of C2 apoprotein, this converts into IDL, and then IDL, if it shed this apoE, it converts into LDL, and LDL will be taken up by LDL receptor that is present on hepatocytes, and through receptor mediated endocytosis will occur and lysis of this all material, and this protein apoB100 will release amino acids will be recycled, and here this fat will also be recycled, or it may be utilized for energy. Now so IDL can also directly uptaken by receptor, especially LRP, LDL receptor related protein. So this IDL will be taken, and this IDL will be taken up by this apoprotein, not by B100. Apoprotein E is very important for IDL. So this is taken up by apoprotein, IDL is taken up by LRP, and it will also go into endocytosis, same process as this LDL receptor LDL will go, same process it will go. So this is the whole process. There is one more important process. So this LDL has one more step. This LDL also takes up HDL, that is, though I have shown HDL here, you have to remember HDL is there in blood stream. So HDL, from HDL, when LDL come into contact in the blood, this HDL will transfer its cholesterol through the protein called cholesterol ester, CE. So this is cholesterol ester. So this cholesterol ester transfer protein, it is associated with this HDL. So HDL, it is present, it is a part of HDL. So this protein will transfer cholesterol ester from HDL to LDL. So this is very important step in HDL metabolism. So how HDL does cholesterol reverse cholesterol transport, in next class we'll talk about cholesterol transport. So HDL will transfer this. So there is one more step in metabolism. After distribution, there will be transfer of cholesterol, acceptance of cholesterol from HDL. So actually for LDL there will be 7 steps and 7 steps. So after shedding, LDL will be formed. LDL will receive protein, this cholesterol ester, and then it will be catabolized. So remember, this is very important step. CETP, cholesterol ester transfer protein. Now this is all about this VLDL metabolism. So VLDL, let me revise in quickly. So first, what will assembly will happen? Assembly, after assembly, secretion of nascent VLDL, and then nascent VLDL will mature after getting 2 extra apoprotein, and then this mature VLDL is ready for metabolism. This mature VLDL will convert into IDL after shedding apoCII, and this IDL will convert into LDL after shedding apoE, and then this LDL will receive cholesterol ester from HDL, and then LDL ultimately will taken up by hepatocyte through LDL receptor. So this is the whole mechanism of this hepatocyte. So the very important point here is how LDL is, where is LDL produced? VLDL is produced. So VLDL is produced in hepatocyte. Second important point is where does it start and where does it end? So it starts in hepatocytes and it will end in hepatocytes. So the third important point is about lipoprotein lipase. What is lipoprotein lipase? Is it activated by which hormone? Insulin activates this lipoprotein lipase, especially adipocyte. Lipoprotein lipase will be activated by insulin, will be induced by insulin. So I'll make a separate video on lipoprotein lipase. There I'll discuss everything about lipoprotein lipase, types of lipoprotein lipase. This is very important enzyme. That is why I'll make separate video on it. Now, so other important is IDL. So IDL is taken up by receptor through E, this apoE. That is why apoE becomes very important. If there is defective apoE, then IDL will increase and chylomicron will increase, because chylomicron remnant, especially chylomicron remnant, and IDL will increase if there is defective E, because this E is the protein, apoprotein, through which it will be uptaken by hepatocyte. This chylomicron and IDL both are uptaken. If this E is defective, they will accumulate, and this will cause type 3 hypertriglycerid, type 3 hyperlipidemia, 3-4 triglyceridemia, hypertriglyceridemia. Type 3 hyperlipidemia is equal to hypertriglyceridemia. So this is all about VLDL, IDL and LDL metabolism. I hope I have covered all important things. So if you have any question, put in the comment below. Please don't forget to subscribe this channel, like this video, and press the bell button so that whenever I upload the video, you will get notification. Thank you for watching.